All right. So welcome to our second TtGG webinar of the 2022-23 academic year. My name is Alexa Narowski, and I am a Genomics Education Fellow working at the Jackson Laboratory in Farmington, Connecticut. Today, I will be presenting a new curriculum module we have developed on sequence comparison and identity. During this webinar, I will discuss the new content that has been developed by our TtGG team, provide a brief introduction to sequence comparison and percent identity calculations, and give you a live demonstration of some of the activities included in our new module. Following this, the TtGG team will go over some program updates. This meeting will be webinar style. If you have any questions or comments, please add them to the chat as they arise. Our team will try to address questions in the chat as we go, and we will address any remaining comments and questions at the end of the presentation. This webinar is being recorded and it will be posted on our website. I will move fairly quickly through the demonstrations, but you will have access to the recording after the webinar if you want to revisit any of the content. Our TtGG team has been designing new lessons and activities in bioinformatics and genomics. The overall goal of this new curriculum is to teach math and data literacy. The lessons and activities will be hosted on our website and will be free and accessible to anyone who wants to use them. We are creating content in 4 different areas, each of which will be its own module. The 4 content areas are cancer genetics, sequence comparison and identity, the region of new variants and mutation in the genome, and ancestry. Our cancer genetics content and the corresponding webinar recording are already available on our website. And today you will hear more about our new sequence comparison and identity module. So let's take a look at an overview of the new module. The sequence comparison and identity module focuses on 3 enduring understandings. First, that changes in the genetic code can affect changes in protein structure, which in turn can change protein function. Second, bioinformatics tools enable us to understand biological data. And third, bioinformatics and genetic information can inform new ideas in a variety of fields, from evolution to human health. The activities in this module all aim to help students address the question, what can we learn from comparing genetic information across individuals and species? And through completing the activities, students will engage and develop a variety of skills, including data analysis and interpretation, computation or math skills, asking questions and formulating hypotheses, problem solving, pattern recognition, and constructing explanations. This is the table of contents from the sequence comparison and identity module teacher guide, so you can see the organization of the module and its components. To start, there is the introductory content, which includes learning outcomes for the module and descriptions of all the lessons and activities. The next section includes all of the materials for each lesson and activity. I'll be giving a demonstration of portions of some of these lessons later in the webinar, but for now I will provide a brief description. The first lesson is a brief reading and knowledge check that introduces students to the concept of sequence comparison and the formula for percent identity calculations. The second lesson is a suite of 5 activities that introduce applications of sequence comparison through the 5 genes from the TtGG laboratory curriculum to give students and teachers an entry point into this sequence comparison and identity module using genes they may already be familiar with. The TtGG curriculum was designed to explore human genetic variation with a focus on variants in 5 different genes: ACE, actinin-3, OXTR, CYP, and TASTER. Each of these genes is associated with an interesting phenotype and has a different type of molecular variant. In this part of the module, there is one version of the activity for each gene, and all 5 of the activities cover the same concepts. Students practice applying sequence comparison and percent identity calculations between individuals, across species, and across gene families. These activities do not require the use of online databases. We unfortunately do not have enough time to cover these activities in detail today, but I would be happy to address questions about them at the end of the presentation. The last set of activities in the module centers around a real research study on a rare neurodevelopmental disorder. First, students will read a scenario that introduces them to some of the participants in the research study and ask the students to put themselves in the shoes of the patients and their family members. Next, in the first full activity in this section, students will use BLAST to identify variants in the patient's DNA that could be causing their disorder. This lesson emphasizes the central dogma, and students use both a codon chart and BLAST to determine how DNA variants impact the amino acid sequences of the corresponding proteins. In the second full activity, students will use bioinformatics tools Polyphene2, UniProt, and BLAST to evaluate the potential impact of one of the patient's DNA variants on protein structure and function. And finally, students will reflect on the guiding questions for the module and dive deeper into related ethics topics, including the outcomes of rare disease research and access to researchers in medical care. So let's explore the module in greater detail. As the students would if you were implementing this module, we'll start with an introduction to sequence comparison. There are two options included in the module for introducing sequence comparison to your students. The first is a reading and associated knowledge check, which you can assign to students to complete either in class or in advance of class. The other alternative is a PowerPoint presentation, which I will walk through now. You're welcome to choose either option or use both. So let's start by defining sequence comparison. Sequence comparison is a technique to determine how similar two or more nucleotide or protein sequences are to each other. To compare sequences, there are 2 to 3 general steps you might follow. The first step is to align the sequences. In sequence alignment, we are generally looking for a way to line up the sequence next to each other such that there are as many matches as possible and as few mismatches or gaps as possible. While we can do this by hand for short sequences, DNA sequences are often hundreds to thousands of nucleotides long. So we instead use computer programs like BLAST, which stands for Basic Local Alignment Search Tool. I will be demonstrating several bioinformatics tools, including BLAST, later in this webinar. The next step is to identify any differences in the sequences. There are 2 main kinds of differences: mismatches, which may represent substitutions, and gaps, which may represent insertions or deletions. When we use BLAST, the matches are displayed as dots in the bottom sequence, while mismatches and gaps are represented as red letters. This format makes it easier to quickly identify the differences between 2 sequences. The last step in this process is to calculate percent identity, which is a quantitative measure of how similar 2 sequences are. We'll talk more about this calculation in a bit. And while this example uses nucleotides, the process is the same for protein sequences. Just instead of a sequence with A's, T's, C's, and G's, we would be comparing sequences of amino acids. To help us understand why sequence comparison can be useful, let's look at an example of an application of sequence comparison: the original identification of SARS-CoV-2, the virus that causes COVID-19. This example is not included in any of the activities of the module, but it provides a current example of how sequence comparison and percent identity calculations are used in real research. When the virus first appeared, researchers took samples from patients with pneumonia of unknown cause. They extracted and sequenced the viral nucleic acid, then compared those sequences to the reference sequences of known viruses. They confirmed that there was no match to a known virus, but this new virus's genome was most similar, at about 85%, to a bat SARS-like coronavirus genome. So the researchers used sequence comparison both to establish the existence of a new virus and to determine where that virus may have come from by identifying its closest evolutionary relatives. Plus, they use percent identity calculation. They specifically state that SARS-CoV-2 genome is more than 85% similar to a bat SARS-like coronavirus genome. How did the researchers come up with that number? Let's look at an example calculation. Here we have an alignment of 60 nucleotides as it would appear on BLAST. The top sequence is the query sequence, or the sequence we're interested in. The bottom sequence is the subject sequence, and in this case, it is the reference sequence, or the sequence we're comparing to. Again, you can see dots in the bottom sequence, which represent nucleotide matches between the two sequences. In this comparison, there are also 6 mismatches. For each mismatch, there is a nucleotide in both the query and subject sequences. There are also 5 gaps, which are represented by dashes. In this case, all 5 gaps are in the query sequence. Together, this makes 11 divergent positions, or places where the new sequences are different. We can now calculate percent identity using this formula. There are a couple of different ways to calculate percent identity, but for the purpose of this module, we will be calculating based on the total number of nucleotide positions in the comparison. For this example, we have 60 total nucleotide positions and 11 divergent positions. If we do the subtraction in the numerator, we get 49 over 60 times 100%. At this point, you can see that what we are actually calculating is the percentage of the total nucleotide positions that are the same between the two sequences. For this comparison, there are 49 matching nucleotide positions and 60 total nucleotide positions. Our formula uses the number of divergent positions instead of the number of matching positions because there are often fewer divergent positions in a sequence comparison, so it'll be easier for students to identify and count them. Following through with this calculation, we get an 81.7% identity for this sequence comparison. So now that you've seen an example calculation, you may be wondering, why calculate percent identity? What does it mean and what might it tell us? Percent identity is a way to quantitatively determine which sequences are most similar across multiple comparisons. In the SARS-CoV-2 example, calculating percent identity enabled the researchers to determine which other virus sequence was most similar to the SARS-CoV-2 sequence. Now that we've gone through an introduction to sequence comparison and percent identity, we can move into a demonstration of some of the activities from the module. In the activity demonstration, I will focus on the sequencing for rare disease diagnosis section of the module, starting with the scenario introduction. This set of activities centers on a phenomena that is based in a real research study and story, which is introduced to students as a scenario. The study started with one family who are the focus of the scenario, but ultimately researchers sequenced the DNA of several patients with a rare neurodevelopmental disorder and published their results in this paper in the New England Journal of Medicine. The scenario takes the perspective of the parent of the first 2 patients in the study. It reads, "You are a parent of 2 daughters, both of whom have an undiagnosed disorder. Your daughters both have similar symptoms, including learning difficulties and ataxia, and an inability to control body movements. You want to find a medical diagnosis for your daughters so that you could connect with other patients and families and learn more about how to best support your daughters. However, you've been to many doctors and none of them have been able to identify a known disorder that matches all of your daughter's symptoms. Finally, one doctor connects your family to a group of researchers. After hearing about your family's efforts to find a diagnosis and care for your daughters, the researchers suggest that they might be able to learn more by sequencing your daughter's DNA. After sequencing, they'll compare your daughter's DNA sequences to a reference sequence to look for any differences or variants. After reading the scenario, students connect this story to their own lives by answering questions like, how does this compare to your experience at the doctor? And what questions do you have for the researchers? This scenario also provides a connection to bioethics, which students have the opportunity to explore in more depth at the end of the module. After reading and reflecting on the scenario, students will write preliminary answers to the 2 guiding questions for the module. The first guiding question is the essential question for the module. What can we learn from comparing genetic information across individuals and species? The second guiding question gets at the enduring understanding that changes in the genetic code can affect changes in protein structure, which can change protein function. Specifically, it asks, how might a DNA variant affect protein sequence structure or function? At this point, students are not expected to be able to fully answer these questions. At the end of each activity, there are opportunities built in for students to revisit these questions and update their answers based on what they've learned. In the first full activity, students follow the researchers' process to learn how they identified a genetic variant that could be contributing to the daughter's disorder. They will also investigate the types of sequences that researchers can compare and discuss why researchers might choose to compare certain types of sequences over others. Specifically, students will use BLAST to align a patient's DNA sequence to the human reference sequence. You may be familiar with using BLAST for sequence comparison or sequence alignment. TtGG has other lessons that utilize this database, and this is going to give you another option for teaching sequence comparison to your students. BLAST is a tool that can be used for several types of analysis. There are options to analyze both DNA and protein sequences, and it is possible to identify a gene or compare gene sequences between different species. In this activity, we will focus on the alignment tool to identify a gene and pinpoint differences between gene sequences. Let's see how this tool can be used to help identify a genetic variant associated with a genetic disorder. So I'm going to switch to my web browser, and I have the BLAST website homepage open. So first, from this main page, we're going to navigate to nucleotide BLAST, which we will use to compare DNA sequences. In the activity scenario, students learned that the researchers sequenced the DNA of the two daughters in order to identify genetic variants. So students will be using BLAST to look at a DNA sequence from one of the patients and compare it to the human reference sequence. For those who aren't familiar, a reference sequence is a DNA or RNA sequence that is assumed by scientists to be a representative example of the genetic material of a specific species. To start, students will input our patient DNA sequence. The patient sequence is called family 2 allele 1, meaning that it represents one of the variants from the second family in the research study. I have all of the sequences that I need in the background, but for the student lessons, the sequences are included for easy copying and pasting. So now I've pasted the family 2 allele 1 DNA sequence in the box under Enter Query Sequence. Next, in this Choose Search Set section, I'm going to type Homo sapiens and tax ID 9606 and select that one into this box next to organism. This tells the software that we are only interested in looking for matching sequences from the human genome. Now I'll scroll to the bottom and click BLAST. When the results appear, students will scroll down to this descriptions table and find this descriptions tab. At this point, the activity asks students to look through the names of the sequences listed and identify which gene the DNA sequence is from. Based on the results here, you can see that all the sequences listed include Homo sapiens autophagy-related 7, ATG7, indicating that the sequence is likely from the human gene ATG7. You may also notice that these sequences are all mRNA sequences. BLAST searches for alignment with both DNA and RNA sequences from sequence databases. Since the patient sequence we are using comes from the coding region of the ATG7 gene, it will also align with the mRNA sequences for ATG7. You may also remember that RNA molecules use uracil instead of thymine. However, most sequence databases use the letter T to represent both thymine and uracil. So mRNA sequences in sequence databases typically contains T's instead of U's, allowing us to align our patient's DNA sequence with the ATG7 mRNA sequence. So next, students will click the Select All box to unselect all of the sequences, and then select only the sequence labeled transcript variant 1 mRNA, since that is what the researchers use as their reference sequence. Now, if I click the Alignments tab, and then in this drop-down menu, select Pairwise with dots for identities, this allows for an easy visualization of any variants in the sequence compared to our selected reference sequence. So in this sequence comparison, this top sequence, the query sequence, is our family 2 allele 1 patient sequence. This bottom subject sequence is the ATG7 reference sequence that we just selected. Looking through the sequences, we can see a mismatch. The query sequence from family 2 has an A, where the subject reference sequence has a G. The numbers at the beginning and end of the subject sequence represent the location of the first and last nucleotide of the alignment within the whole sequence. So if we were to count over from the left, we would find that this mismatch occurs at nucleotide position 1771 in the reference sequence. So now we know that our patient has a variant in their ATG7 gene, but we don't yet know whether that variant is contributing to their disorder. The next thing we will check is whether this variant impacts the amino acid sequence of the ATG7 protein. The activity first asks students to do this using a codon chart, and then has them check their answer using BLAST. In BLAST, we can check the box labeled CDS feature up here. This feature identifies the coding region and translates both the reference and query sequence into a protein sequence. In designing this lesson, we selected a sequence that was in frame with the coding sequence, and such that the first nucleotide in this sequence here would align with the first nucleotide of the codon. Now, if we look at our sequences, we can see that the variant changes one amino acid from an arginine, as we see in the subject reference sequence, to a histidine, as we see in the query patient sequence at the top. And again, if we count it over from this left side, this first amino acid is number 572 in the sequence. We can see that our variant occurs at amino acid number 576 in the protein sequence. Now, after establishing that the variant does have an effect on the protein sequence, students will calculate the percent identity for the sequence comparison. We can see here that BLAST calculates a percent identity, which is labeled identities, up here at the top of the alignment. For this comparison, the percent identity is 98%. However, this calculation is only for the 60 nucleotides in this short comparison, as we can see from this fraction 59/60 here. In reality, the ATG7 gene is much longer than 60 nucleotides. So next, we have the students do some of their own percent identity calculations. Students will recalculate percent identity for the patient's ATG7 sequences using 4 different sequence types: the whole gene, the mRNA sequence, the protein coding sequence, and the protein amino acid sequence. Students will then be prompted to select which of these comparisons and percent identity calculations provide the most useful information about this particular variant. So in that first part of the activity, students used BLAST to identify the protein change and calculated percent identity for a missense variant, which causes substitution of one amino acid for another. In part 2 of this activity, students look at another patient's DNA sequence, which has a nonsense variant in ATG7. Nonsense variants change a codon that codes for an amino acid into a stop codon, producing a shortened protein. In part 3, students investigate a third patient variant, which at first glance appears to be a missense variant. However, the variant actually changes the way that the mRNA is processed, resulting in one exon getting left out of the final RNA. This causes a shift in the coding sequence, again producing a shortened protein. In part 4, students look at an example of a silent variant, or a DNA variant that does not impact the amino acid sequence. This final variant is not from a patient sequence, but it provides an important counterexample to the other 3 variants that all do affect the amino acid sequence. At the end of the activity, students are asked to reflect on the different ways in which DNA variants can impact amino acid sequences. Students will also reflect on which type of comparison and percent identity calculation they identified as most useful for each of the different variants, whether their choice was the same across all of the variants, and if not, which comparison type they would choose to be consistent across all of the patient variants. So to recap, in the first activity, students used sequence comparison to identify a specific gene in which the patients have variants and to determine the effect of those variants on the amino acid sequence of the corresponding protein. So they learned about how, in the research study, The researchers identified variants in the patient's DNA, specifically in a gene called autophagy-related 7. They also learned that the patient's DNA variants result in changes in the amino acid sequence of the patient's ATG7 proteins. Specifically, from part 1 of the first activity, they learned that family 2 allele 1 results in a change from arginine to histidine at amino acid number 576. This variant will be the focus of the second activity in this section. While students can learn this information from the first activity, we do also provide this background information at the beginning of the second activity, so that you can have students begin with that second activity if you wanted to use it independently. So now let's move on to the second activity. In this activity, students will use a variety of bioinformatics tools to investigate whether the patient's ATG7 variants have an impact on their ATG7 protein structure and function. Once the students have the background information about the research study and the family 2 allele 1 ATG7 variant, we ask them to reflect on what they know about the relationship between protein structure and function to make a prediction about whether the patient variants will impact protein function. Next, we'll give them the tools to better evaluate whether the family 2 variant will affect protein function. First, students will use a tool called polyphen2 to predict whether the family 2 variant in the ATG7 gene will affect ATG7 protein function. Polyphen2 stands for polymorphism phenotyping version 2. It is a tool which predicts the possible impact of an amino acid substitution on the function of a human protein. Let's take a look at how polyphen2 works. So this is the home page of polyphen2, and this main page of the site has a submission form in the middle here that can be used to submit a query about a specific protein and variant. We want to use polyphen2 to predict whether the variant in family 2 allele 1 is likely to have an effect on ATG7 protein function. So in this field here, typed protein or SNP identifier, I will enter ATG7 underscore human in all caps. Since I entered a protein name, we can skip the next section that asks for the protein sequence because polyphen2 will pull the sequence from an online database based on the identifier I just provided. Now we know from the first activity that the DNA variant in family 2 allele 1 results in a change from arginine to histidine at amino acid number 576 in the protein sequence. Students will use this information to fill out the next several sections of the form. First, in the field titled position, students will enter 576. The next two sections list all of the amino acids as single letter codes. In the section labeled AA1, students will select R for arginine. This represents that the amino acid in the reference sequence is an arginine. Then students will select H for histidine in the section labeled AA2. This represents that the variant amino acid in the patient sequence is a histidine. In the field titled Query Description, students can enter a name for this submission. I will use family 2 allele 1. Then we'll click the button labeled Submit Query. When this page loads, we can see a list of completed and pending submissions, also called jobs. You can see the family 2 allele 1 submission under the pending jobs. I'll wait for a minute to give the tool some time to process our query, and then I'll click this button down here labeled Refresh. There is now a link labeled View in the results column next to our family 2 allele 1 submission. I'll click on that link to view our results. Once we have the results page open, we can find this results section and specifically the prediction confidence report. Here we can see that polyphen2 predicts that this patient variant is probably damaging to protein function. At this point, students are likely to be wondering, how did polyphen2 make this prediction? At the end of this part of the activity, we have students make some hypotheses about what information polyphen2 could be using to make this variant effect prediction. Then in the next two parts of the activity, we use additional bioinformatics tools to show them the types of information that polythene 2 uses and why that information is useful. So in the second part of this activity, students will use UniProt to gather more information about the function and structure of ATG7. UniProt stands for Universal Protein Resource. It is a database which contains data on protein sequences and functions for a wide variety of organisms, including humans. First, I'll navigate to the UniProt entry for human ATG7. This entry is linked directly in the activity, but students can also search the database to find the correct entry. At the top of the entry is some basic information about the protein, including its name, its length, and the gene from which it comes, or the gene which codes for it. The first section of the entry, down here, contains a summary of the protein's function. Students are guided to this section and provided questions to help them interpret the section, including what are some science words you recognize, what are some science words that are unfamiliar to you, what type of protein is ATG7, and ATG7 is involved in a process called autophagy. What do you think autophagy means? The goal of these questions is to have students discover that ATG7 is an enzyme and that it is involved in autophagy. We also hope students will realize that they are able to recognize and understand some of the science words in a heavily jargon-filled text. Next, students will look at the features section of the UniProt entry, which is right here. This section contains information about important structural and functional features of the protein. Here we can see that the active site of ATG7 is at amino acid number 572, which is rather close to our variant at amino acid number 576. The activity prompts students to think about how an amino acid change close to the enzyme's active site might affect protein function. So as we just saw, in the second part of this activity, students read about ATG7 protein function on UniProt. And after investigating structure and function on UniProt, the students will be guided through evaluating simplified 3D structural models and looking at the amino acid properties of arginine and histidine to infer how the patient variant might affect protein function. In the third part of the activity, students will look at whether the family 2 variant is in a conserved sequence of the protein. For those who are not familiar, a conserved sequence is a protein or DNA sequence that is identical or highly similar across multiple species. If a region of a protein is conserved across many species, we can often assume that the structure of the region is important for proper protein function. We will have students investigate this using BLAST by comparing the human ATG7 protein sequence to sequences from mouse, chimpanzee, yeast, fruit fly, and chicken. Before beginning, students predict which of the 5 species are most and least closely related to humans. Then, students make predictions about percent identity for these ATG7 protein comparisons. Specifically, which species they expect to have the greatest percent identity and what percent they expect that to be, as well as which species they expect to have the lowest percent identity and what percent they expect that to be. Next, students check their predictions by identifying the percent identity for human ATG7 compared to each of the 5 species and filling out this data table using BLAST. Then students will compare a sequence of 50 amino acids around the site of the patient variant and calculate percent identity for just that region. By comparing the percent identity for the region of the protein near the variant site to the percent identity for the whole protein, students can determine whether the variant is in a conserved region of the protein. Let's take a look at this in BLAST now. For this alignment, or for this activity, students will use Protein BLAST. So I'm going to click on Protein BLAST here. First, students will select this option here to align 2 or more sequences. So we want to make sure that's checked. Then in the box labeled Enter Query Sequence, students will paste the human TtG7 protein sequence. As before, I have all the sequences that I need in the background, but for your students, the sequences that they need are all included in the lesson for easy copying and pasting. So now I have the human TtG7 protein sequence here. Oh, and let's try that again. There's the other box. Okay, so next we're going to paste the chimpanzee, mouse, chicken, fruit fly, and yeast sequences in this Enter Subject Sequences box. You can go ahead, we can copy and paste all 5 sequences simultaneously. And if I scroll through here kind of slowly, you can see there's fruit fly, mouse, chimpanzee. So we have all of our sequences pasted in this box. Now I'm going to go ahead and click Blast. So now if we scroll down, we can see that all 5 of our comparisons are listed in this table. Students will then navigate to the Alignments tab here. And in the Alignment View dropdown, we're going to select Pairs-Wise with dots for identities. And now we can scroll down the page to look at each comparison. Within each comparison, the query sequence is the human sequence, and the subject sequence is either the chimpanzee, mouse, chicken, fruit fly, or yeast sequence. As we scroll, you may notice that the sequences further down, which are the fruit fly and yeast sequences, have a lot more red, representing more amino acid positions that are different from the human reference sequence. To get more quantitative evidence for this observation, we can locate the percent identity score for each comparison, which is the value labeled identities at the top of each comparison. Here we can see that yeast has a 38% identity, fruit fly has 41%, chicken has 84%, mouse has 93%, and chimpanzee has 99%. At this point, the activity prompts students to revisit their predictions about which organism would have the highest and lowest percent identity to check whether their predictions were correct. Next, to see if the patient variant is located in a region that is conserved, we'll look at the percent identity of a 50 amino acid region around the site of the variant. The region we have selected is approximately 25 amino acids on either side of the variant. So now I'll copy the sequence of 50 amino acid, which is also included in the lesson document for your students, and search for it on the BLAST site. Searching for the sequence should highlight the region of interest in all 5 comparisons, as you can see here in the chimpanzee comparison. Students will then calculate the percent identity of the 50 amino acid region for each comparison. As an example, let's look at the comparison for human and chicken TtG7. Here we can see that in this highlighted region, in the comparison of human and chicken TtG7, there's only one divergent amino acid in the comparison between human and chicken, which is this one right here. Since we are looking at a 50 amino acid region, this corresponds to a percent identity of 98%. And if we look back at the percent identity for the whole protein, we can see that that value is lower at 84%. If we were to repeat this for each of the comparisons, we would find that, except for the chimpanzee comparison, the percent identity of the region around the variant is higher than the percent identity for the whole protein. Let's take a look at this data summarized in the table from the activity. Looking at this table, we can now clearly see that for most of the comparisons, the percent identity values for the region around the variant, which are in the rightmost column, are higher than the percent identity values for the whole protein, which are in the middle column. The only exception is the chimpanzee, but the percent identity values for both chimpanzee comparisons are quite high, only changing very slightly from 99% for the whole protein to 98% for the variant region. To conclude this part of the activity, we ask students to determine whether the variant occurs in a conserved region of the protein, using the data they've just collected in this table to justify their answer. So in this activity, we've used Polythene2 to make a prediction, then used UniProt and BLAST to gather more information about ATG7 and the location of the variant within the protein structure. In the final part of this activity, students will reflect on what they learned in parts 2 and 3. They will use what they learned to update their answers regarding whether the patient's variants likely impacted ATG7 protein function, and what information the Polythene2 model could be using to predict the effect of a variant on protein function. Finally, students have the opportunity to complete a final reflection on the guiding questions. Additionally, as one of the core strands of the TtGG program is bioethics, we have also developed bioethics questions to accompany this section of the module. Many of you are aware of our collaboration with the Personal Genetics Education Project through Harvard, called PGEd. In our PD programs, we highlight some of the lessons that PGEd has developed on personal genetics. For the sequence comparison module, we've written additional discussion prompts and questions emphasizing the same bioethics framework. The first half of the bioethics content asks students to consider whether there is value in knowing the genetic cause of a disorder when there is no treatment available yet, and to think about having a conclusive diagnosis might impact patients and their families. This content expands on an existing PGEd snapshot on identifying and understanding rare genetic conditions. The second half of the bioethics content focuses on the question, who has access to researchers' genome sequencing and therapeutics? Specifically, students learn more about the patients included in the study and consider what barriers may exist for other patients to access genome sequencing for diagnostic purposes. This content expands on an existing PGEd snapshot on the cost of therapeutics for spinal muscular atrophy. Now that you've seen the activities from the module in more detail, let's look at some of the resources available for implementing these activities in your classroom. In addition to student copies of each lesson, we have also developed a full teacher guide with a clickable table of contents that you have been seeing throughout this presentation. This guide includes all the lessons and activities with discussion notes. We have also provided a few example implementation strategies and outlined NGSS alignments for each activity. There are also links to supporting materials, which include PowerPoint slides, tutorials, and additional related ethics lessons from PGEd. Of course, the way you incorporate this material in your classroom is completely up to you. We've provided activities of different lengths and difficulties so that you can find specific sections that could work with your existing curriculum. Activities from this module could align well with existing curriculum on the central dogma, evolutionary relationships, and the focus on a protein involved in autophagy could align with a cell biology unit. Alternatively, if you wanted to use several of the activities and lessons, you could develop this into a sequence comparison unit. There are also answer keys for all lessons, and these are available upon request. As I just mentioned, to offer support for both teachers and students, we've generated a series of tutorials for the databases used in our activities. These tutorials demonstrate how to perform specific functions in each database. Links to specific tutorials are embedded into the lesson, including the student versions for quick access. There are also YouTube playlists of short videos demonstrating different functions of BLAST on the JAX YouTube channel. Additional video tutorials for Polythene2 and UniProt are both planned for development in the near future. This sequence comparison content is live and available for immediate use. As of now, the module is available on the Genetics Learning Resources page of our website. You can access this page by visiting the website listed here or using this QR code. The teacher guide and student versions of each activity are all formatted as PDFs and are available to download for printing or distribution. And as mentioned previously, there are answer keys available by request by emailing the TtGG team. Remember, you can also always email us at ttgg@jax.org with questions about this content or anything else about the TtGG program. This concludes the demonstration of our new sequence comparison and identity module. I can address questions shortly, but first I'm going to turn it over to Christina to provide program updates.
Thank you, Alexa. We have just a couple updates to share in case you haven't seen our prior announcements. We are collaborating with Lab Exchange to put a lot of our resources and create new online digital resources onto the Lab Exchange platform. Lab Exchange, if you're not familiar, is an online learning classroom that has tons of different resources from simulations and interactive activities to standalone lessons and activities. All of our content is currently there. And again, we are building new ones, including, next slide, please, Alexa, one that we have already built, which is a gel electrophoresis simulation. So this is a fully immersive virtual simulation of a lab environment for gel electrophoresis. This follows the TtGG ACE gene protocol, but of course can be used for any sort of learning experience around gel electrophoresis. It is a really great way to teach some concepts around this laboratory technique if you're doing the TtGG protocols, good supplement to working in the lab. It's also useful even if you haven't been able to get into the lab this year, you don't have your own equipment or don't have any sort of access to laboratory techniques and programs like gel electrophoresis. So do check it out. And we have more simulations and interactive activities to come that will be announced in future program updates. Finally, we do want to share that 2 more modules for TtGG are in development. 2 of them have been released, including this one, and 2 more coming in the new year. One on new variant mutation, origin of new variants and mutations in the genome in March. If you have been following, this is a new date that we have, so just make sure that you keep up to date with all of our future communications to show this date change. As well as the final one about ancestry coming later in May. And just a reminder that this webinar module that you just watched is available online, as well as our previous one on cancer genetics. That's also available on our website, and the webinar introducing the activities and lessons are also on YouTube. And with that, we thank you so much for your attention and welcome any sort of questions.
Thank you very much. I just need to run and go get my daughter. Sorry, but I appreciate everything.
Thanks for coming.
Thanks for coming.
Thanks for coming.
Beth.
Well,